So this is a lesson on genetic engineering for A-level. Okay, so I'm going to start with what you know from GCSE. So the idea here that we're going to get bacteria to produce human insulin. So we get the idea that we cut the gene that codes for the insulin from the pancreas cell using an enzyme called a restriction enzyme. You get the plasmid from the bacterium and you cut it with the same restriction enzyme. More about that in a moment. You're then able to join the two pieces of DNA together using a ligase enzyme and put it back into the bacterium, which will then reproduce and produce human insulin for you. As we'll see at A-level, that's not quite the whole truth, so there's quite a bit more detail for you here. Right, so back to the bacterium. So we have the plasmid, which is just represented here, and the main circular loop of DNA is there. So the plasmid is removed from the bacterium, can be as simple as putting it into a blender, and then the plasmid is there are lots of different plasmids. Think of them like tools for genetic engineers. This is a commonly used one, and it's very useful because it's got antibiotic resistance genes, which allows you to only grow bacteria that have successfully taken up the plasmid. Because if you put them on you plate them out onto a agar containing the antibiotic, then only the ones that have taken up this plasmid will be able to grow on it because they've got the resistance genes. The millions of bacteria that haven't taken up the plasmid because your technique wasn't successful would be killed. It also contains sites for cutting it open with restriction enzymes, and these are special enzymes that cut DNA when they recognize a specific sequence. So two different ones here, and we've made two different cuts, but as you can see we've got single-stranded pieces of DNA on the end, and these are really useful because they're what they're called sticky ends, which is a rather sensible name for once in biology. We don't have many of those. And the idea is if you use the same enzyme to cut open your human DNA and your plasmid DNA, then you'll have the right shaped sticky ends for the enzyme to be able to join them together again. And this particular plasmid has got lots of different sites, so it's easy to cut open and insert whatever gene you want there. Right, so the obvious place to start is the pancreas, because this is it's only in these cells that the human insulin gene will be switched on. Okay, so, but if you remember from what we did earlier on this year, it's a bit, no, sorry, this is last year, wasn't it? Introns and that. It's a bit more complicated than that. It's not as simple as just cutting out the human insulin gene, because as well as cutting out the coding regions, you'll also cut out the junk as well. These non-coding regions aren't translated into a polypeptide. So what you have to do, you have to wait until it's been transcribed into mRNA. After the mRNA has been spliced, so the introns are removed out before translation, so then this is the really useful bit. This is just what the mRNA that will be translated by the ribosomes, so no junk DNA in there at all. So, but we've got a problem in that this is RNA and not DNA. So, thankfully we have the retroviruses. Unfortunately, there's one particular retrovirus at the moment we're not very thankful for, but these viruses have an enzyme called reverse transcriptase, which takes a copy of the mRNA and makes a single-stranded piece of DNA, which is called copy DNA or cDNA. So once you've got this piece of cDNA, then you can just use your standard DNA polymerase to turn that single-stranded piece of DNA into a double-stranded piece of DNA. So this is exactly what you want. You've got no introns within it, and you've got two-stranded, and it's DNA. Now, if you've cut it with the right restriction enzyme, the same one that you used for the plasmid, then you can insert it straight into the plasmid. using a ligase enzyme. So that's one thing we told you the truth about at GCSE. Then it's just a case of getting the plasmid back into the bacterium, allowing the bacterium to reproduce, and it will then produce the antibiotic for you, and you can extract it, purify it, and there's your product. So this can obviously be done with any gene, really, in theory anyway. So that's what we've looked at here. It could also be used in the process of replacing faulty genes in terms of gene therapy, and what we're doing more and more of now, particularly with the CRISPR, the really precise genetic engineering, so precise that it's called gene editing nowadays, then we can add these beneficial characteristics to our crops, etc. Right, so it's just a case of reading all of this through, really. I will talk you through this and the PCR, but the main thing I need you to do, really, is to complete the tasks at the end, and then I'll put some past paper questions up for you as well.